In today's class, we will study clinical trial and RCT. What it mean by clinical trial? The clinical trials is nothing but if any new drug is found, that new or any new device or new method of treatment, if you want to analyze, then we are using the human volunteers. In that, the new drugs we are doing the trial. This is the called as a clinical trial. Now the RCT, the randomly we are allocated the participants in the groups, that is the RCT or randomized controlled trial. That mainly two groups we are dividing. One is the interventional group, in that we are giving the new drug, and the another is control group, in that the placebo, it may be placebo, it may be standard drug, like that we are dividing. Okay, now when we will study one by one. Before that, this topic comes under the BAMS syllabus, third phase. In the third phase, research methodology subject, the fifth chapter and sixth topic is clinical trial. What it means by clinical trial? The clinical trial is one of the research study, in that the systematic research study is conducted on human participants, using the human participant to evaluate the safety, efficacy, effectiveness of maybe new drug or devices or the procedure or interventions. For that, we are doing this clinical trial. The next one is the RCT. In short, RCT, it is called randomized clinical trial. Why it is called randomized clinical trial? The RCT is mainly, we are two groups. In that, we are using the two groups. One is the interventional group. Another one is the control group. In that, randomly the participants we are allocating. That's why it is called randomized controlled trial. It means that randomly the participants we are selecting in any group, and control means in that along with your new interventional group or interventional drug group, along with that another group we are taking, that is called controlled group. In that, we are doing the trials on the humans. This is called randomized controlled trial. In simple meaning is that randomly the participants, human participants, you are using in the two groups. One is the interventional group, another one is the control groups. This is the RCT. This is considered as one of the gold standard in the medical research, because this is everyone is accepting it is one of the genuine one or reliable research, because the randomly we are allocating the participant in this study. Now the definition of the RCT: types of clinical trial in which it is one of the type of clinical trial in which the participants are randomly assigned into two or more groups, in interventional group and control group, to compare the outcome and reduce the bias. What it means in this is one of the clinical trial. It comes under the one of the clinical trial in which the participants are randomly we are allocated. It may be due to like that cheat methods, the selecting this one and another method, the computer itself is randomly they are dividing and selecting, or randomly we are selecting. Any one methods we are using, lottery method also we are selecting to in the separating the groups, the participants assigned in the two groups. Mainly two groups we are using. One group we are calling as a interventional groups. Interventional group is nothing but the new trial drugs, in that groups we are using. And another one is the control group. The one is the interventional, another is control group. The control group, in that it may be the placebo, it means that sugar pill or saline injections we are giving, and or else we are giving the standard injections or standard medicines. It means the two groups mainly we are doing using in the randomized clinical trial. In that, we are comparing here. What we are doing in that, we are comparing the outcomes. What is the outcome? Why we are doing? Because to reduce the bias, to reduce the one-sided result. That's why we are doing the RCT. That's why definition is this is one of the type of clinical trial in which the participants are randomly assigned into two or more groups, and interventional groups and control groups, to compare the outcome and reduce bias. Okay, the next is the key features. What are things are there in this RCT? The first one, it is a prospective design. Prospective design means from the today onwards, in the futures, in the next coming days, we are doing research. This is the prospective research. Retrospective means past history. We are not doing here retro. The prospective means the next coming days you are doing the research. The prospective design, it is the randomized allocation of the subject. It means that randomly we are selecting. Just now I told randomly, it may be lottery methods, it may be computerly we are dividing the subjects, like that. It may be allocation randomly. Use of control groups. Just now I told the mainly two groups we are dividing. One is the interventional, another one is the control group. In the control group, it may be placebo or standard treatment we are giving. Next one is the blinding. Okay, blinding is nothing but we are hiding the what in the medicine it is there. Okay, we are not giving the telling the participants or the doctors which in which group we are giving the which drugs. This is the blinding, hiding the medicines. It may be three types. It is a three types: single blind, double blind, and triple blinds. Single blind means it means that the participants don't know whether in which group they are in that which group they are involved. And second thing is the participant don't know which medicines they are taking, whether it may be sugar pill they are taking, the placebo it may be, or it may be standard drug they are taking, or it may be interventional drug they are taking. The participant only don't know. The single is nothing but the only participant don't know. Double blind is nothing but the double blind means the participant don't know in which group, and as well as doctor don't know which in that capsule or tablet which is the what is called the genuine medicines, the interventional drug it is there, or sugar pill. That is a double blind. Triple blind, it means that the participant as well as doctor as well as the who is entering the data, that is data analyzer, also don't know. All three peoples don't know the what is that in the groups. That is the triple blind method. The next one, measure cause and effect. Why we are doing the RCT? The RCT is doing because the drug is whether effective or not in the particular treatment. It means that the cause effect relation, it is there or not, whether drug is effective or not. For checking this trial we are doing. This is the measure of cause and effective relation. The next third one is the phase of the clinical trial. The mainly four phases we are using in this clinical trial. While we are doing the clinical trials, the four phases in that we are doing the clinical trial. First phase is the test safety and dosage on the small group of the volunteers, healthy volunteers. Whether the drug is, we are tested on the preclinical trial on the animals, whether this drug is safe or not in the human volunteers. To check the healthy volunteer, first thing is the testing. Only few people we are taking, less than 100 people we are using, healthy volunteers we are taking. And dosage, for the first phase we are using for the fixing of the dose. Small dose we are giving and increasing the dosage fixing purpose. The second phase, in that the number of the volunteers we are increasing. So first phase, if you are taking less than 100, in that the more than 100, 200, 300, 400, up to that you can take in the second phase. Assess the efficacy and side effect. In the second groups, suppose if you are taken 300 participants, it means that here we are checking whether drug is effective or not, or the side effect is produced due to if you are increasing the peoples. That also we are assessing. Third phase we are using for the comparison purpose. In these groups, again you are increasing the number of the participants. Suppose in the 300 if you are taken in the second phase, now you can increase up to 2000, 3000. Suppose 1000 if you are giving the interventional drug, it means the new drug, and one is the control drug. That we are comparing and we are analyzing that. Compare new treatment with standard treatment in large group, whether the drug is effective or not. That we are checking in the third phase. Now the fourth phase is the post market. It means that government is approved your drug, and next the drug is came to market. After the market, it is available to the common peoples. Then after that, is there any long term side effect produced? That also we are observing in the fourth phase. This is called post-marketing study to monitor the long-term effects. This is the fourth phase. Okay, this is about the phases. Next one is the advantages. What is the advantages of RCT? The first one is the minimize the bias through randomization. If you are randomizing the randomly, the participant don't know in which group they are, the bias we can minimize. The next one, provide high level evidence of effectiveness, that we can get the evidence of the as well as the effectiveness of the drugs. The next one is the ensure scientific reliability. Because of this scientific method, the everyone is believed that is a reliable, because of you are doing the RCT, ensure the scientific reliability to control setting. In that, if you are doing control setting, that everyone is reliable. Next one is the limitation. What are the limitations of this RCT? The first one is expensive and time consuming, because it is a costly, because so many patients you are changing time and regularly doing, that's so expensive, as well as time consuming. It may take 10 years, 14 years, 15 years. That time consuming, it is there, may involves ethical issues, because the randomly you are doing some things, the ethical issues it may comes. Next one is limited general ability due to strict selection of criteria. It may select, while you are selecting the participant, you are randomly selecting, it may be due to that the some problem may arise. This is the limitations of this of the RCT. The next one, very good example of the COVID. Okay, in the COVID pandemic, what the in the Covaxin as well as Covishield vaccines, while they are preparing in the pharmaceutical companies, they also did the RCT. Okay, how they did? During the COVID-19 pandemic, were conducted to evaluate the efficacy of vaccine like Covaxin and Covishield. Participants were randomly divided into two groups. One received the vaccines, while another received the placebo. It may be saline injections or distilled water, like that they received. The outcome, the protections against the COVID, it is there, or side effects were then compared to determine the vaccine is efficacy, it is there, safety it is there. Based on that, they are proved. It means that in the COVID pandemic also, the RCT used to prove this their vaccines. Okay, this is about the RCT as well as the clinical trial. Hope you understood our today's topic. Okay, if you understood, please share to others, like, share and subscribe. Thank you.